Okay. Hello, everyone. Thank you so much for joining the webinar. My name is Danielle. I'm with Proteintech, and we're joined here today by Dr. Kimberley Clark. And she's going to be talking about a lot of really interesting topics. And before we get started, I would like to just briefly introduce her. Dr. Kimberley Clark is a postdoctoral research fellow within the Sweeney Oncotherapeutics and Biology Laboratory at the South Australian Immunogenomics Center Institute, or Cancer Institute, at Adelaide University. Dr. Clark gained her PhD within the myeloma research laboratory before undertaking a successful postdoctoral position in the cell signaling laboratory. Throughout her studies, Dr. Clark has been interested in the molecular mechanisms that underpin cancer biology and novel therapeutic targets in not only multiple myeloma, but prostate and breast cancer. Her current research utilizes biologically relevant tumor models, in vitro and in vivo, to explore novel treatment approaches for treatment-resistant lung cancer. So thank you so much for joining us and talking a little bit more about the work you're doing. And with that, I can hand it over to you. Just as a reminder for the attendees before you get started, we will be kind of watching the chat. You're welcome to share any questions you have in the chat. We will address some of those questions at the end and try and answer as many questions as we can get through. But without further ado, Dr. Clark.
Easy. Thank you so much for having me. It's a pleasure to be able to talk with you all today. And so I'll be sharing a bit of my experience in optimizing 3D cancer models for our various projects. And I do want to caveat, I do not feel like I'm necessarily an expert in this, but I think that I have some useful things to share that, you know, if others are also troubleshooting and trying to develop these models, that hopefully they might be some just things to avoid and things to maybe try for your own kind of uses in the lab as well. And I'm still very much applying a lot of these techniques and optimizing them as we go, which I think is something that we are probably continually doing in this field. And so just to outline briefly a few things that I will touch on today, I'll be exploring the different models that we've been utilizing in our lab, how we've generated these and how they kind of compare for answering different research questions, a little bit about some of the troubleshooting that we've experienced and how we've applied immunofluorescent staining in these models. And lastly, how kind of Proteintech's been able to assist in this optimization step to enable better fluorescent antibody labeling in our 3D models. And Danielle did a fantastic introduction for me, but I just wanted to kind of quickly touch on, I guess, my research journey so that it provides some context of kind of where I am in the research landscape. So I'm based in South Australia, and I did my undergrad at Flinders University before completing a PhD at the newly opened SAMRI as the only kind of institute in South Australia at that time. And then I transitioned to Monash University in Melbourne, where I completed a postdoc under Roger Daly in the cell signalling laboratory. And then really during that time, Sargency and the research landscape in Adelaide was taking off, and I transitioned back home to Adelaide and have been with the Sweeney Laboratory for several years now, where we largely sit within the resistance theme of research and explore the molecular mechanisms underpinning resistance to many different cancer therapies. And in particular, my focus is in lung cancer at this moment. And so just to touch on that a little bit more, in the past, I've explored the role of the tumour microenvironment broadly in cancer initiation and progression, and drug resistance is really where my research interests lie now. And for lung cancer, we're exploring the role of stress, treatment-induced stress, and the signalling networks that modulate that response to treatment. Because largely with lung cancer, we see that while patients are diagnosed and often they can have targeted therapies available, and that can be initially quite successful, resistance ultimately develops in most patients. And we really believe that a large part of that is this kind of intrinsic balance between the state of the cell stress and pro-survival signalling, and that's where our research questions seek to answer that, and where we are applying these different 3D models. And so I guess I just wanted to start on kind of why we're exploring 3D models of cancer. I think for me especially, my background had largely been looking at in vivo models where I've done a lot of preclinical mouse work in the past, and it's been in this recent postdoc that we've largely transitioned a lot of this work into 3D models, and that's for a number of different reasons. Probably the most critical reason for this transition is that the pipeline from basic research to approved drugs, it kind of has this critical funnel. A lot of that is due to identifying the correct molecular compounds and targets, but there's also this key cutoff point around this kind of between stage 3 and I guess approved drugs, where a number of drugs that show great promise in preclinical research fail to transition into the clinic, and ultimately kind of over 90% of drugs that do even reach these initial stages of clinical trials are not successful due to a number of reasons. And so we do need to identify drugs better, and I think we've identified that in vivo models are not necessarily the way to do that. And that really takes me to the second point, is because often what we're seeing in mice and where we have these fantastic results, we've cured cancer. I think, you often probably have seen those different sites that are purporting to have cured cancer in mice. It's not biologically relevant when we transition that to humans, and that's something that we can actually utilize a bit better in 3D models because we can apply human tissues. And increasingly, we're seeing that funding is kind of relying on the better application of 3D models and more biologically relevant models. As we've seen from the European funding bodies recently, there's targeted consideration that non-animal models are utilised more effectively in research. And so this is something that is going to be critical for many of us as we generate different projects. And of course, that also leads into there's many ethical considerations. You can replicate things a lot more efficiently in 3D models. And if they're biologically relevant, it raises the question of why a lot of this is being done in animal models. And also cost. You can replicate things and work a lot more efficiently in a number of these different 3D settings. And so I think the most important thing when you're starting to develop your 3D models is kind of what is the research question you want to answer, because there are a number of different approaches that can be taken. And we actually use a combination of pretty much all of these approaches to answer different aspects of our research questions. And for me, that research question is largely at the moment characterising treatment-naive and treatment-resistant non-small cell lung cancer populations. And we characterise these from a number of different ways. We can be looking at how they respond to different drugs and combinations of drugs, how their cell morphology and behaviour changes, and how they kind of interact with other cell populations. And so I think I wanted to just start off that we still definitely use 2D culture models. This is a strong basis for where we start a lot of our research. It's easy, it's efficient, we can establish cell culture very effectively here. And we often use it as our starting point to test initial research questions where we are not then moving on to more labour-intensive techniques. And it's also fantastic where we can generate some genetically modified cell populations with a lot more ease. And we can, if we're using cell lines, we can maintain continual culture a lot more efficiently. And I'm going to touch on these next 3 models in a bit more detail, but I just thought I'd introduce them here. But we've been then expanding on these 2D culture models with 3 different approaches to 3D cancer modelling. The first is spheroids or tumoroids, they're called in it sometimes. We've been using cell lines in mono and multicellular populations to generate spheroids in ultra-low adherent plates, and then we've been able to assess those in a number of different ways. And these have been quite readily generated in our non-small cell lung cancer populations, but have required different optimization for different cancer types where we see varying levels of success. And we've utilised this as a model to determine changes in cell growth and drug response, and we noticed that drug sensitivity across from 2D to spheroids drastically changes. And we've expanded upon the spheroid setting into growing cells in 3D matrices to generate kind of individual spheroids. And we're using a system from rastrum for this, and that we can use that to assess cell growth. And this model is a bit more beneficial for also exploring migration in the presence of drugs as well. And it also enables live cell imaging over time, where we can monitor the change of the cells and how they're interacting in the 3D space over a longer time period that is not so accessible in the spheroids. And finally, one that we're working closely with Sylvia at our MiniWell is the BioTwin chip. We're very much still optimizing this, but I did want to highlight it today because it's been a critically useful model. And I think while we haven't utilized its full potential yet, it's one that we're definitely still working on, and I think is the closest one to the biologically relevant models we need. And this has enabled us to explore kind of cell-cell interactions with a little bit more finesse. We can really control how we want the cells to be plated and interacted with a lot more precision. And we can assess drug penetration and response a lot more effectively in this model as well. And so I'll just touch on how we've optimized and developed these models. And then I've got a couple of kind of case examples of some troubleshooting we've done for each of these as well. So in our laboratory for spheroid generation, this was our very first model that we tried for 3D cell growth, and we took a very simplistic approach using ultra-low adherent plates. And for this, we have generated some beautiful results in kind of a very simple model, and I think has been a great starting point for our exploration of 3D cell culture. So for this, we've utilised either monoculture or combination culture of non-small cell lung cancer cell lines, and often combining that with lung fibroblasts as a cell line as well. And the different numbers and cell ratios for these have had to be individually optimized for the kind of best generation of spheroids and how long we can culture them for. So that'll be very cell type specific. But for us, we can combine those, spin them down, and within a week you have some beautiful spheroids that are then able to be treated over kind of another week or so. And we can get some really nice imaging and analysis from that point onwards. And an example of a very initial basic type of analysis we've initially looked at is just the spheroid shape that these cells take. And so what we'd noticed in 2D culture from a very early time point when we generated resistant cell lines is that the morphology of the cells differed within 24 hours of treatment, and that morphology change was maintained over the subsequent months of treatment. And we wanted to compare how that looked in our 3D models. And we did see that our treatment-naive cells formed kind of quite a bulky core of cells with only minimal aggregated cells around the edge, whereas our resistant cells to a common EGFR tyrosine kinase inhibitor osimertinib formed a much less bulky core and had a lot more dispersed cell aggregates that were around that. And that's something that we could quite readily quantify and has been just a really nice starting point for how we've explored our 3D cell growth. And I will touch on this a little bit more on how we've furthered that, but our next step of analysis, often for the spheroids, while we have been able to image them as entire 3D kind of complex models, we had found that often they don't retain their shape very well in this particular one. There's no matrix for these cells. And so where we've been able to get our best results is actually sectioning them. We embed the spheroids into OCT and cryosection them. And then we can look at sequential sections on slides and look at a number of different stains and get really high resolution immunofluorescent imaging to look at then the different interactions within the center of the spheroid from that point onwards. And so then introducing the next 3D model that we've been utilising is the rastrum. We've been really lucky at Sargency to partner with Rastrum and to be getting their lovely pink bioprinter, which has got the image at the top here. So it's a really funky little machine. And what we can do here is when you can order specialized matrices to combine with your cells where you're getting 3D cell growth within a matrix, but it's a lot more customizable than what we've experienced with just using maybe base matrigel or other hydrogel bases. And so what we can see is we're readily able to tune the matrix stiffness. And this has been important as we look at different cancer types. We've been branching out into exploring pancreatic cancer, which is known as a very kind of ECM-rich cancer, often quite stiff. And when we're looking at matrigel, the stiffness of matrigel is kind of sitting in that 30 to 70 Pascal range, whereas in many cancers, you're looking in the kind of in the thousands, and that's just not something that can be achieved with traditional matrigel very easily. And so in this setup, we've been able to optimize the different matrix stiffness really quite easily And we can also select different matrix components to answer kind of or suit specific biological questions. So for lung cancer, we've been able to include kind of different amino acid chains that are relevant to the growth of our lung cancer cells, such as collagen type 1 and fibronectin. And also we can explore multiple formats for printing these. So we've largely been using a 96-well plate format because it enables us to do a lot of replicates quite easily, but you can also use larger well formats and kind of mixed matrix models as well, which is something we're yet to explore, but I think provides lots of nice options for easy setup of 3D cells. And so this is kind of what over the course of about 5 days, we can see the growth of this particular cell line in one of the less stiff matrices, but we can quickly see by day 5 we're getting these nice kind of independent growth clusters coming from the original single cells that were seeded within the matrix on day 0. And kind of as we optimize these things, we've been able to compare how the cells interact between different stiffnesses of matrices. So comparing on the left-hand side a less stiff with a more stiff matrix, can see that the morphology and the spread of the cells varies quite differently. And in this, we've got cells that are just treatment naive, and then those treated with osimertinib, our kind of drug of choice for many of these models. And we can see that the area, when we analyze these using the IncuCyte live cell analysis, varies quite differently and gives us slightly different results. So it's something that is been considered quite strongly, and then where we go from here and which is the most optimal plate format going forward. But we have been able to see kind of a nice transition from what we see in 2D culture, where we can answer some key research questions if our inhibitor of choice. We haven't published this work yet, so I'm keeping it kind of relatively de-identified, but the use of the rastrum has enabled us to model cell growth over 7 days, and then we've treated them with osimertinib or in combination with our kind of test inhibitor. We can see that the double combination has resulted in a significant reduction in the total area of the cells in this 3D matrix. And then we're able to image them using immunofluorescence for markers of cell death and apoptosis. And also, if your cells are fluorescently labelled, you can track these over time as well. So a really useful addition. The one caveat being you need to use optical plates for this so that you can get the best images, as just using standard cell culture plates won't give you the best results. And then the third different model that I wanted to share that we've been using today is the AminiWell BioTwin chip. And this is the kind of the most structured system that we've been applying, and we've been getting some great hands-on advice from Sylvia from AminiWell. And this is a chip that sits on essentially the same slide as a microscope slide. So it is really readily available for imaging. And it enables us to answer kind of all of the questions we need. So you can investigate cell migration, drug penetration, and readily investigate the interaction between different cell populations. So you can see the chip layout on the left-hand side. It kind of comprises of 4 sections, and so each section you can use and repeat your experiment or test with different drugs. And so if I'm not sure if you can see my pointer, sorry, I'll just grab the pointer up. Sorry, never mind. So on the kind of zoomed in portion here, you can see that there's a center ECM well separated with 2 outer wells that you would put your cells of choice. And this has been great for us because as we're investigating how our tumor cells interact with our other populations, one of the things we've encountered is that sometimes it's you've got competing cell growth profiles of these, and by mixing them together, you start to get a dominant population, and you can't accurately assess how they interact with each other over time. So this enables us to control that a little bit more tightly, where we can either seed one population first, followed by a second, or you can separate them across the ECM matrix, and so that they can still signal to each other, but are not necessarily out-competing each other for cell growth. And so you can still answer your research questions without having to be monitoring different populations. And While there are many other organ-on-a-chip methods, a lot of those utilize specialized hydrofluidic systems that can be quite costly, and they require kind of very specialized setups, whereas we've found the benefit of using this chip system is that it's a standard plate format, and apart from requiring some hydrogels to seed in the middle and the plates themselves, they're otherwise quite ready to go. And for us using the BioTwin chip, this is just a bit of a close-up of what our cells have looked like growing in the chip over time. We've been able to kind of do a fibroblast base layer where we can seed the fibroblasts first and then seed in the epithelial tumour population after that. And so we can kind of create this encapsulated cell population and that then expands into the centre matrix. This has really been useful to try and mimic something that might be seen within the tumour microenvironment, where these cells are often existing within this kind of supportive core of fibroblasts. And like I mentioned before, we can also then put them across the channels from each other so that they can talk to each other and then interact within the centre channel as well. And so now that I've introduced some of the different models that we've been utilising, I wanted to touch on some of the different ways we've been analysing them and some of the troubleshooting that we've been experiencing as well. So one of the key things that we've experienced is that immunofluorescent antibody staining in 3D models doesn't readily translate from our 2D model systems. So one of the key considerations is that these can be slightly more sensitive to long wash times, and especially in our spheroid models, we were seeing a loss of tissue architecture as we were doing repeated washes, and as well as staining time, where you're needing extra time for the antibody to penetrate into the centre tissue or through any matrix that you might have used. And these were really steps that required significant optimization and kind of was where we often fell down and when we initially started trying to use our existing workflows for immunofluorescent staining. And so a couple of case examples, and this one is maybe less about the immunofluorescent staining and more about kind of finding something interesting along the way, is when we were looking at our non-small cell lung cancer spheroids. So when we section these spheroids, we get these lovely cross sections through the middle. When you're just looking at DAPI, you can see you get a nice even section of cells right through the center of the spheroid. Something that looks quite beautiful. If we're looking at then cell-specific markers, in this case, we're looking at a keratin staining. You can see again that that's quite nice across the entire cross-section of our spheroid. Whereas when we came to then, we had a fibroblast marker on our green channel antibody, and we found that in our non-stained control, we were getting a lot of background staining. And this was quite bright. It's something that surprised a lot of the team, and we couldn't quite find an answer for why we were getting this significant background staining when we had no secondary kind of fluorophore. This had nothing that should be expressing green except for autofluorescence. And even then, the autofluorescence was not consistent across the entire tissue. We were getting quite unique bright peaks in different cell populations. And so we wanted to see maybe if we could utilize this to actually answer a research question rather than throw out these slides and start again, or use nothing in the green channel. And after a little bit of digging, what we kind of did come across is that we could potentially utilize this green signal as a measure of a very specific type of cell death with FAD. And it's been published quite extensively as a marker of cell death. And we actually transitioned from utilizing just our standard lab microscope to the confocal LSM 980 that we have down in Adelaide microscopy. And what we can do there is look across the entire emission spectrum and look more specifically to confirm that this was a type of fluorescence coming specifically from FAD, which we did confirm. And so we've actually utilised this in a workflow to complement our other methods of analysing cell death, and are using this autofluorescence to help identify dead cells within these particular spheroid populations. We can see that we get a bulk of these dead cells within the centre of the spheroid, which we would expect as we often have a more apoptotic or necrotic core. and that it's kind of quite specific in certain cell populations, and we don't often get a strong overlap with DAPI either. And so what we've been able to do is when we analyze our 2D cells looking at different drug treatments, we can see we get a separation. We're basically looking to see that our combination treatment is more effective than our individual treatments alone. In 2D culture, that looks great. It's very easy to assess, and we can use caspase to analyze that. And what we've been able to show is that when we translated this into the 3D spheroid cultures, we could actually use this autofluorescence as a measure of that, and it nicely aligns with what we were seeing in the 2D cell culture. We have confirmed this utilizing the caspase signaling and also other dyes such as cytox that are more specific for cell death. But it's been a really nice workflow we've been able to use where we haven't been just, like I mentioned before, been able to throw out this result. We've been able to pivot and find something a little bit unique. And so given that we are in a Proteintech webinar, it would be remiss of me not to kind of touch on how we've been able to work with Proteintech and their systems to benefit these workflows. And so as we were first approaching the development of these 3D models, we did struggle with the fact that a lot of the antibodies that we have in the lab are largely multi-purpose. We are using the same antibodies across Western blotting, and then we're also wanting to use those for immunofluorescence and other uses as well. And most of our antibodies that we have are rabbit, because we work with a lot of human cell lines, and rabbits are commonly a common species across most of our antibodies. And we didn't want to be buying new antibodies that are directly conjugated or multiple different species so that we could multiplex our antibodies. And this led us to exploring the flexible system with Proteintech, where we could directly conjugate our existing antibodies with their system, and that enabled us to be able to multiplex the antibodies. It enabled us to use the same species and our same existing antibodies that we have used and trust without having to increase kind of staining or wash times through sequential stains. And we've found that this maintains a nice high fluorescent signal. We've actually been applying it through a couple of different projects now where we've got kind of closely related antibody staining happening across breast and prostate cancer projects, and have been kind of really happy with the results so far. And one thing that has been particularly useful for us is the use of the flexible calculator, because initially when we started applying this, suddenly the mental maths really expands, and you're trying to figure out kind of how to approach the different antibody doses and how to conjugate them in the right volumes with your fluorescent linkers. And so this has been a tool online that's been invaluable for that. And I think as new students have come onto our team and we've been kind of teaching them the different techniques of the lab, this has been a key resource we've been able to show to them to say, if you're trying a new antibody and want to be linking it, this is your first port of call. And this is just a nice example of how we've been able to apply the flexible system in our 3D models. So we now are Manuel set up where we've got the central ECM matrix and an initial channel where the cells sit. It was really important to us that we could identify different cell populations going through the channel and determining where our fibroblast populations were growing and where our epithelial populations were growing. So the first thing we wanted to do is optimize that we could identify these different populations. And so we started by trying to identify our fibroblasts. We were using a fibroblast-specific antibody, FAP, and we conjugated that with the Correlate 488 flexible antibody. And we could see then some really nice distinction throughout the more crowded initial well, and you could see that infiltrating into that central ECM layer as well. And so this gave us more confidence in both the model and our ability to stain these cells. We did need to increase the staining time in this particular 3D model kind of beyond 1 day, like a standard overnight stain, to a couple of days. But we are quite happy that we're now getting nice staining of our particular cell populations. On the next slide, a slightly less good image because I actually had some trouble with our microscope. So this was just on our upright rather than on our confocal. But when we combine that with the cytokeratin antibodies, we can at least see that we can identify both our fibroblast and epithelial populations quite nicely within the well, and then how they infiltrate down into the central matrix. And so I feel like I might have sped through this a little bit, but it does leave us some time for questions. But I just wanted to summarize kind of where our different models have been utilised and some of the pros and cons for each of those, and how that might come into the consideration when you're choosing which models to start with or to answer the next research questions. So we kind of use the spheroids in an ultra-low adherent plate as a good starting point. Very simple setup, very cost effective, only needing the kind of U-bottom plates. And once we could stain and section those, we were getting really high quality immunofluorescent images across the spheroid cross-section. However, this does require additional time to embed and section the spheroids. We are only getting one spheroid per well, and there's no matrix interaction. And so kind of you have to weigh those up against each other. When we transition to the rastrum cell printing, this is enabled in a very high throughput format, and having multiple 96-well plates printed with only a couple of hours enabled us to test multiple different drug conditions quite quickly. And you can stain within the plate, which maintains that 3D architecture really nicely. However, we have found that the matrices from rastrum can interfere with the background signal, and so we have had to work with kind of making sure we've got the best antibodies and staining times to avoid having the matrix interaction in those as well. And then lastly, for the biotwin chip from a mini well, this has given us the most complex interactions to investigate, has been great for cell migration, and the imaging is very easy to get high quality imaging because it's already on a nice glass bottom slide. This time consuming, the setup for this one has been slightly more time consuming because it requires a couple of steps to seed out the different components, and is something that we require some more optimization with, but I think probably provides us the most biologically relevant model across all of them. But in reality, what we've been doing is using a combination of all the different models, and we complement that with our existing 2D culture, as well as some in vivo work as well. But largely, this is where we're using our most biologically relevant translational work to provide to pharma companies and our other collaborators as we look at different novel drug combinations. And so I'm going to leave it there and just allow some time for a bit of questions and anything else. But otherwise, thanks for listening to my experiences, and hopefully that can be useful in some way going forward.
Thank you so much, Dr. Clark. I will keep an eye out for any questions either in the chat or the Q&A. Looks like we have one coming in now. The question is, how exactly does Flexible allow multiplexing of antibodies? Does this involve primaries of the same species?
Yes, so definitely. That's where we have been able to use primaries of the same species. So I think everything that we've used so far has used rabbit primary antibodies. And then we've been able to directly link the antibodies with the fluorescent linker. And so you have separate tubes where you can link your antibodies within 10 minutes. And then once they're linked, you can quench that signal and combine them together into a single kind of master mix. And that way that master mix can just go straight onto your kind of model or slide of choice, which has meant that our workflow is really straightforward, really simple. We don't have to do sequential staining over days. We don't have to wash off antibodies between and worry about that so much. We're getting still really specific antibody staining with the same species in one mix.
What is the typical plex for some of these applications? How many targets or colors are you working with at one time?
At the moment, we've been working with up to 4 colors. And that is including DAPI or kind of another stain in the background there. But we've been working up to 3 colors. But I think the sky's the limit, really. There's a number of other color options. For us, we've been largely using it for identification of cell populations, and then the addition of, say, an apoptotic or cell growth marker. So we've tried it with Ki67 and different caspase antibodies as well. And so that's enabled us to just ask some really nice, straightforward questions where we can say, kind of, if we've got a mixed model, what are the cell populations? Where are they? And are we getting a preferential response to any drug treatment from one population or another? Because a lot of the time we're wanting to see, are we seeing kind of tumor-specific cell death, which we haven't been able to look at kind of in a broader sense, because usually you're just getting, say cell-targeted glow or other systems where you say, okay, we've got less cells or we've got more cells. We want to be able to say our drug treatment is working in a more tumor-specific manner, and the multiplexing allows that.
Great. Got a few more questions coming in. There's one on the autofluorescence signal you got in the 488 channel, how you managed to utilize it as a measure of dead or dying cells. They went on to say organoids in their hands autofluoresce in a different channel, more the 555 or 594 color channel. Do you observe autofluorescence in different channels when you look at different types of spheroids?
That's a really good question. We haven't observed a lot of autofluorescence in those channels. Whether that's we're often getting quite a strong, I'm going to come from a different way. We haven't observed a lot of autofluorescence in those other channels at all. And I think that's what made this finding quite striking, because it was so bright that it didn't take on the appearance of what we would consider kind of general autofluorescence, or that kind of really broad, non-specific autofluorescence, and is then something that, because we could specifically look at that across the entire emission spectrum and say with confidence that this was coming from this FAD feature, that it was a feature of cell death. I guess I probably don't know enough about other forms of cell death or autofluorescence that might be coming up at those other spectra. But there's definitely a wealth of literature about utilizing autofluorescence in this manner, but it does at this stage seem to be something that's coming from those kind of mitochondrial stress features, which is what we're detecting in ours. But yeah, who's to say that it's not a biological feature from the other channels?
Yeah, very interesting. Another question asks, what media combination worked best for fibroblasts and epithelial co-cultures that you showed in the Omini well?
Yep. So it depends how you want to set up in, I guess, a mixed culture system. So when we have a epithelial layer and a fibroblast layer across the two channels separated by the ECM, you can actually use your specific cell media to each cell type, which is a nice feature of this setup, so that you don't have to kind of be putting one of your cell types in a media that is less ideal for that. In the mixed systems, we had previously optimized and observed we could culture our fibroblasts in our tumor cell media. So that's going to differ depending on what your base medias are, but we approached that by making sure that each cell population could be maintained in that media for at least a few days to a week at a time in the kind of complementary media. And if they couldn't, then we looked at kind of what the key, I guess, ingredients were across the two, and determined from there if there was a kind of a third media that was more optimal. But I think that's where having those two channels separated really helps, because you can keep each population as happy as possible, which has been one of the key things that has been hard looking at the combination 3D cultures, because often you do have to sacrifice the kind of happiness, so to speak, of one cell type for the other. And then you may be interfering with the results you see because you're stressing out as one population rather than maintaining them in their peak state.
Makes sense. Another question asks, what is the optimal size of spheroids for a drug efficacy study?
Yeah. So again, this is very depending on the cell types that we've been using. For the lung cancer models, we've been seeding about a combination of 5,000 tumor cells to about 4,000 of our spheroids, because I think the next question I saw was the ratio of the fibroblasts, so I might as well do a 2-for-1 for this one. So I think what we found for our lung and fibroblasts was, about 5,000 of the tumour cells for 4,000 of our fibroblasts. And we found then that over about 5 days of culture, we were getting a kind of a solid condensed spheroid. We can actually essentially pick them up out of the wells at the end. So they do form quite a solid spheroid. However, when we looked at that in breast cancer cells, we don't see the same kind of morphology come through, and we've had to pull that cell number right back. So it depends on the proliferation rate of both of those cell types. And I think we've erred on the side of slightly smaller spheroids often because we then do find that the wells don't support the size of them after a while, and you see that they get a lot more cell aggregates around the edge where it seems like they can't be supported in that central spheroid. And so we've been I guess is one of the key considerations is that we want to see most of the cells forming that kind of spheroid rather than floating around the edge and not kind of interacting in that same way.
Great. Thanks for answering both questions at once. There's also a question about how to get started with setting up flexible multiplexing in a lab.
So I think I would start from the point of if you have a workflow that is working really nicely for any staining protocol you currently have. So in our case, we have tried and true antibodies that have come from, our different postdocs to different labs. I would start using your tried and true antibodies. And then if you're already having to conjugate those, it's a really simple kind of swap out where you can then go online and look at the flexible calculator to see how much you might need. And it essentially just replaces your secondary. And so it removes that step entirely. You can directly conjugate it and you're proceeding like you have a primary antibody that is directly conjugated. So I think, I would start from a trusted antibody and then go from there.
Great. I'm glad it's been working so well for you guys.
Yeah, I think it's been nice to have something that it gives us a bit more confidence as we've been able to kind of stain with multiple targets at a time. And the time consideration, I think we're all pretty time poor. Looking at a multi-day staining protocol when you're trying to fit it into an already busy week can be a bit of a headache. And so having something that you can just kind of add directly in and get results essentially, the next day or the next 2 days has been a big win.
Great. That's good to hear. Another question for you. We can probably fit a few more in. You mentioned having to incubate your antibodies for 1 to 2 days for good staining with the OminiWell platform. Is this due to the thickness of the 3D culture or the matrix?
It's probably due to both. I think also because the setup, we've got channels and then a central well. And so when the antibodies go through, we need time for that to penetrate through both the cells and then into the ECM. So you're going to have probably a condensed amount of cells existing within the channels, and they are within matrix as well. And then you're having a central matrix core. So because you're wanting to get very consistent staining throughout all portions of that, if you're letting it stain for less time than kind of those couple of days, what we were seeing initially is bright staining of our kind of top populations of the wells, and then kind of a lack of staining as we got into the ECM. But we knew there were cells there because we could visualize them through phase contrast. And so we really needed that extra time for it to settle through kind of both the cell thickness and the ECM. So yeah, it's a bit of both. And I think you can even extend that out to, 3 days or more. For that particular system was the one that needed the longest staining time.
Great. And I think that leads us really nicely into the next question, which is, are there any challenges associated with permeabilizing samples of different thicknesses for the immunostaining?
Yeah, that's a really good consideration. I think for the ImmunoWell, we didn't have to permeabilize that too much. It was just a matter of time, and we just found that time was kind of the critical factor there. For the rastrum, where we've got cells sitting in probably a much stiffer gel than the other systems. I think for those, again, time was really just the critical factor, and it's probably, I think it's going to be very context dependent on what your 3D setup is. Probably the one that we've had the most trouble permeabilizing is when we've tried to look at the spheroid as a 3D spheroid. And I think we found that we were not getting the best antibody penetration through that. And I don't have an answer for that one. Unfortunately, we didn't find the ideal solution to permeabilize the 3D organoid, and that's probably why we transitioned to cryosectioning and looking at it as a cross section, because we just couldn't get the best staining throughout the tissue.
Great. Thank you for answering so many questions. I'm glad we could squeeze so many in here at the end. I think we can probably wrap up the Q&A session. I wanted to remind the attendees that we will be sending out a link afterwards to the webinar, so you can go back, rewatch the webinar as needed. And Dr. Kimberley Clark, thank you so much for joining us. It was very informative. And thank you so much for taking the time to speak with us and the attendees today.
It's been my pleasure, and I'm always happy to make new connections. So if anyone ever wants to chat further, then my email is always open.
Fantastic. Great. Well, thank you everyone for joining us, and we'll see you hopefully on a future webinar as well. Take care. Bye.
Thanks.